Everyone. My name is Ismail Abdullah. I'm a fourth-year medical student. And inshallah, uh, by the end of today, first of all, can you guys hear me clearly?
Yes.
Okay, good. Like, inshallah, by the let me just sorry, hold on. Okay. So, inshallah, by the end of the day, I would be going over most of OSPE, if not all. Inshallah, by the end of the session, you can master OSPE. So if you have any questions, just feel free to interrupt me in the middle, or you can type it out in the chat. But it would be easier if you can just unmute yourselves and let me know if you have a question. All right, bismillah. Let's start. So just to make sure. Okay. But so basically, I divided my presentation to four different disciplines: basic laboratory, microbiology, pharma, immuno. Now, from basic laboratory, you can expect to see 1 to 3 questions. Most of the chunk is going to be from micro. Usually, they ask one pharma question a year, and like same goes with immuno. And I'll make sure to cover all the questions that have been asked in the past in my presentation. So let's start with basic laboratory. So it's all about staining. Now, the reason why we stain is to be able to visualize and, assess morphologies of different cells, different organisms. And we have two ways to stain, depending on the purpose. So do you just want to see microorganisms growing on a dish, or do you want to be able to differentiate and be able to diagnose what kind of organism is growing? Okay. So we have a simple stain and we have a differential stain. Simple ones are just to see that, okay, there's actually bacteria growing in this stain. So we have a positive and a negative kinds of stain. The positive stain is just a simple one. So as you can see here, let me just change my pointer. So this is simple staining. It's just going to show you that there are, you know, certain organisms growing in there. Okay. Negative staining, usually they use something known as the Indiana ink. Okay. This ink is gonna give you these white areas that are not stained. Basically, this indicates that there's, growth happening there. There's an organism right there. In a differential stain, however, it's gonna differentiate it for you. It's gonna tell you that there's this certain organism and this certain other organism growing up. And this is what we mostly use in practice to be able to differentiate certain organisms, like to be able to understand the difference between Staph aureus, um, Staph epidermidis, and etc. All right? Like, uh, I just want to make sure that I'm being heard clearly, and if my camera is not lagging or something.
شوف الكاميرا بتلاقيني بس صوت عادي.
Okay, it's fine, I guess. Come on. So for the differential stains, we have two stains that we need to work through. We have the Gram stain and we have the Zeal-Nelson stain. And طبعا other ones that I'm going to discuss inshallah throughout the session. So let's start. Like in Gram stain, first of all, we use it for which organisms?
بكتيريا.
Yep, we use it for bacteria. Okay, specifically for bacteria. Now, حلاوة Gram stain is it will help us to differentiate between those bacteria who have thick cell walls and those who have thin cell walls. Hello? So let's take a look. So how would they ask you in the exam? They would ask you first of all, what's the differentiating step? They might ask you, uh, how can we stay stated? And I'm gonna answer those questions for you. So first of all, we apply the dye, which is just simple crystal violet dye. Tamam. And then to fixate this dye, okay, to make sure that it's genuinely bound to the cell wall and it's not going to be washed off, we add iodine. So it's going to form these crystal violet iodine complexes. Actually, 2 years ago, they asked us this question in MCQ, so it's pretty high yield. Okay, so this is the fixating stage. It helps you to fixate it. okay? Because then when you add alcohol to it, it's gonna wash away, okay, the remains of the remains of the dye. Those who have thin cell walls are not gonna be stained purple, all right? And then we will add another counter stain. So those who were not stained will be pink, all right? So we add saffron, and it's gonna give you this pink color right there. Well, what's your takeaway from this slide? Just a minute. It's giving me my internet connection is unstable, so I'm just gonna edit. Um, I'm just gonna do one thing. Just give me a minute. So is my camera still lagging or is it good?
No, not as much.
But I hope it's okay then. So I'm just gonna keep going. Um, all right. So start moving on. So the takeaway from this slide is the takeaway from this slide is that the critical step, the differentiating step when the gram stain is the alcohol, the decolorization process. All right. Because this will help us in, differentiating between those who have thick and thin cell walls. So it doesn't just come down to that. There's so many other things, but this is the basics. Hello? And those who are gram positive are going to be purple, and those who are gram negative, they're going to be pink. Tamam. Moving on. So we have certain organisms that are acid fast. So what does it mean, acid fast? Does anyone know what acid organism? All right. So acid fast organisms are those organisms that will not be able to be decolorized when you add alcohol to them. So if you go back to this slide, okay, you put crystal violet, you put iodine, all good, but when you put alcohol, they're not going to be decolorized. So this is a problem. Tamam? So for these organisms, we use specific stain called the Zeal-Nelson stain. And this is high yield. Like this usually pops up. If not in your OSPE, it's gonna pop up in your MCQs. Okay, so the only difference is that you add this thing called carbofuscin, and then you will fixate it by heating it up, and then you will decolorize and add a counter stain, as simple as that. But you need to know why is it, you need to understand the nature behind acid-fast organisms. The thing is, they have a very thick waxy layer, and this makes it difficult for them to be, just stained as we do for other, like gram-positive and gram-negative organisms. And these organisms are Mycoplasma and Nococardia. هلا طبعا في كتير, but these are the main ones that are very high yield, and these are the ones that were in your slides. So you want to know them, okay? So it's Mycoplasma and Nococardia species. And this is how these are your organisms here, and this is how they're properly stained. Them all. So now let's move on to types of media. Now this is uh, genuinely important because these terminologies are going to keep popping up again and again. So I need your undivided attention here. So let's start with base media. So this is general media. Where we're in agar, you're gonna find with just simple nutrient required for growth. But
Um,
sometimes certain
sorry, Sorry to bother you, but I think
Uh,
it's lagging a little bit.
my video or my voice? What exactly? Or
I
everything?
think
Every,
both.
everything is lagging, yes.
Everything is like, okay, how about you give me a minute? Just give me 1 minute. Okay, can you guys hear me?
Yes.
All right, but I think it's gonna be better now, inshallah. Let me retry. Okay, how is it? Am I not lagging anymore? Is everything good?
Your voice is clear, but your image is lagging a bit.
Oh, it's fine. Not about damage. Okay, so again, types of media. So let's start with the simple nutrient agar. This is just a normal routine agar you're gonna find in any microbiology lab. It just has these nutrients, very simple nutrients required for growth. Okay, like not much. But sometimes some organisms require, you know, require specific conditions to be able to grow. Like some organisms need blood to be able to grow. So such medias are called enriched media. Like we add these extra specific nutrients to help and facilitate the growth of certain organisms. Hello, everything's good so far? So now we move to the selective media. When we say selective in microbiology, we mean that there's only one organism that's going to be able to grow on this media. So when I say one organism, I mean like one species of organisms is going to be able to grow on a selective media. And the rest are going to die. There's like, it's not allowed. It's just one only. While in differential media, multiple organisms of the same species, of course, can grow, okay, but they would appear, you know, differently. Like, they would, for example, in this example, I bet you know this, the alpha and the beta hemolysis, right? So like this appears to be different than this one, for example, and this can help me differentiate between these organisms. And the best example I could ever think of would be uh, the mannitol salt agar. So this is a very high yield slide. This was asked, I think, in my batch, and I think as well, like maybe in 12th batch as well. So and also in 13th batch. By the way, guys, I'm 14th batch. Yeah, I'm that old. Um, so here, okay, we have this thing called the mannitol salt agar. Now, only staph species are going to be able to grow here. Why? Because of the very high sodium chloride levels in this agar. But then these staph species, okay, some of them are going to ferment mannitol. So mannitol is a type of sugar, okay? Some organisms are going to ferment it and some are not. So if you take a look, for example, here, Staph aureus is going to ferment mannitol. So when I say ferment mannitol, it's going to utilize mannitol, all right? And the color is going to change from pink to yellow. While Staph epidermidis is just going to remain remain pink because it's not going to use mannitol. So in this way, I have selectively allowed only Staph species to grow here, and now I can differentiate between them, Staph aureus and Staph epidermidis. So is this concept clear? Because this is like 80% gonna show up. So is this clear to you guys?
Yes.
Okay, it does. Moving on. So on streaking technique, this is a very low yield. I don't think it's honestly gonna come in the exam, but just for sake of inclusion, make sure that when you're streaking, you streak evenly, okay, in 4 sets of streaks as shown here, and you streak evenly among the entire agar, okay, so that all you want to make sure that as much of your colony is being exposed to the nutrients in the agar. Tamam. Moving on. So some microbes require certain growth requirements. Okay. And now mainly is because the kind of enzymes they have. So for example, we have aerobic microbes, meaning that they can grow at normal atmospheric oxygen, right? Why is because they have these enzymes called the superoxide mutase and catalase. Now, these enzymes are crucial, of course, because uh, oxygen reacts with cells, okay, and this is gonna cause ROS, reactive oxygen species. And this is going to increase the oxidative stress that might actually result in cell death. Okay. So when we have these enzymes that are going to tackle down the ROS species, so they're going to tackle down the oxidative stress. Okay. That's why they're able to grow, in oxygen. All right. Um, And then we have anaerobes. Anaerobes do not have any of these enzymes. And because they do not have these enzymes, they're going to die instantly, or they're just not going to be able to grow at oxygen levels. Facultative anaerobic, yani, they can grow in both oxygen or no oxygen, like they're they're facultative. Lastly, we have the microaerophilic. Now, microaerophilic has only the superoxide mutase. Now, how the question would come to you, it would tell you that, oh, an organism was growing, okay? This, as soon as we started to keep increasing the oxygen levels, growth stopped. This is a microaerophilic organism, tamam? Why were they able to grow at very low oxygen levels? Because they have the superoxide mutase, but they do not have the catalase. They need both enzymes to function and be able to grow without any stunts. Okay, moving on. So those who cannot live or cannot grow in oxygen levels, okay, we need certain media for them to be able to grow. So we have the thioglycolate broth and we have the cooked meat broth. You only need to know how they look and you only need to know their names. That's it. وطبعا there are specific organisms that grow in specific broths. Um, okay, so yeah, but so far you just need to know that these are the medias needed for anaerobic organisms to grow. And this one is just it's just controlling the environment, try and make sure that there is no oxygen in there, okay, just to facilitate anaerobic conditions. Some organisms are capnophilic. So when we say capnophilic, we mean that they love to grow in very high levels of carbon dioxide. And for that to happen, we would, put them in like a enclosed jar where there's a candle. A candle, basically what it does is that it's going to take oxygen, combust oxygen, and deprive the jar of oxygen and release carbon dioxide. So this is how it really works. Very simple, very chill. I don't think this is going, but just to let you know. Play. So before we proceed, let's just have a quick checkpoint to make sure you guys are still with me. Uh, so can you tell me what's the name of this stain?
Nelson.
Zeal Nelson. Okay, great. Play. Can you name for me the organisms that could grow on this stain?
Mycobacterium tuberculosis.
Okay, cool. Did everybody else get that? Come on, let's move on. Play. This is another agar. So can someone tell me what's the name of this agar?
Manitou fault.
mannitol salt agar. Hello. And can you tell me why is it both selective and differential?
Selective because of the NaCl, and differential because it can differentiate between the different types of staph by mannitol.
Yep, exactly. Mumtaz. Cool. So let's keep going. And the differentiating step in gram staining,
Yeah.
that would be alcohol. Very good, because this is the step where we decolorize. Hello. And so this is a question. It says that during an experiment, microbiologists observed that certain bacterial strains survive brief exposure to oxygen, but struggle with prolonged exposure. Which of the following choices identifies the bacterium?
B.
So the B. So it's microaerophilic because it has only the superoxide dismutase. Tamam. The key word is surviving brief exposure to oxygen, but then they struggle, to keep going. Tamam. So now we're done with basic laboratory, moving on to microbiology. So it's going to be a very long one. So let's start. But so gram-positive, hello, they're going to appear purple on stain. And we have two uh two species that you want to be very familiar with, Staphylococcus and Streptococcus. Okay. How can we differentiate between them? The trick to differentiate between them is to undergo a catalase test. Tamam? When you undergo a catalase test, what happens is that if it's if it's Staphylococcus species, they're gonna show up a bit of bubbles. It's gonna be fizzy. But if it's Streptococcus, okay, it's gonna be just still, no bubbles. Hello? Like, so now, in Staphylococcus species, there are two very important species you want to know, Staph aureus and Staph epidermidis. Tamam? And these are four different tests that I'm going to go through thoroughly, inshallah, to differentiate between Staph aureus and epidermidis. طيب. In Streptococcus, you have these four organisms that you must know. A lot of questions come on these organisms, honestly. And we have three different tests that we use to differentiate between them. The sheep blood agar, presence of certain antibiotics, and lastly, the CAM test, which I'm going to go through, inshallah. So the takeaway from this slide, catalase test will help me to differentiate between staph and strep. Staph is going to be positive, strep is going to be negative. Yeah. So let's start with the tests that we use to differentiate between Staph aureus and Staph epidermidis. So the first one is coagulase test. Now, uh, actually, when I think of Staph aureus, okay, Staph aureus, is like a guy who comes with aura, and he's gold, okay, and he's gonna be a very strong guy, tamam. So he has everything. So coagulase test is gonna be positive, DNA test is gonna be positive, even their appearance on mannitol is goldish. Okay, so this is how I really think about it. So if you look here, for example, in coagulase test, Staph aureus is going to be positive. Okay, it will have the ability to coagulate. Staph epidermidis is going to be negative. Hello. In DNA's test, okay, Staph aureus is going to be positive. So it has the ability to break down the DNA. And as you can see here, this is a strip of DNA, and around it, the mom, you can see that there is like signs of hemolysis, so a breakdown of DNA. Tamam. This indicates that this is Staph aureus. This is positive. This is no DNAs, so it cannot break down the DNA. And so this is Staph epidermidis. Tamam. So moving on, we have the mannitol salt agar, which I believe I already discussed. So Staph aureus is gonna ferment mannitol, so it's gonna turn from pink to yellow or gold. The epidermidis is just gonna remain pink. And on blood agar, Staph aureus is going to be, again, goldish appearance. These colonies are going to be goldish. Staph epidermidis is just going to have white colonies. Tamam? So, takeaway as a summary overall, Staph aureus is going to be positive for everything. It's going to be positive for catalase. It's going to be positive for DNAs and coagulase. and it's going to appear goldish in both blood agar and mannitol salt agar. Staph epidermidis is just going to appear white on blood agar, and on Staph epidermidis is just going to remain pink because it does not ferment mannitol. Okay, so we're done with staph. Any questions about staph species? Or like the tests we do for them? But uh, now let's move on to Streptococcus. Tamam. So the very first thing we do is sheep's blood agar. This is a very important test because it differentiates them. Hello. Now, when we add those Streptococcus organisms on a blood agar, tamam, what happens is, the way they're gonna break down the blood cells is different. So some we're gonna have partial hemolysis. Partial hemolysis, they're gonna appear just a bit green, okay? Beta hemolysis, they're gonna appear very bright because they fully hemolyze the blood there, not partially, unlike alpha. Gamma hemolysis, there is no hemolysis. So those who are under the category of alpha hemolysis are two: strep pneumonia and strep viridans. And those under beta hemolysis are strep pyogen and strep agalacti. Tamam? And now we're going to differentiate between each. So let's start with the alpha group. Alpha group, as I mentioned, we have pneumonia and we have viridans. The very first test you want to do is the optogen test, which is just a kind of like an antibiotic, actually. Okay. And you want to see if if they are susceptible or resistant to it. So pneumonia is actually going to be susceptible because see, there will be no growth around the antibiotic disc right here. There is no growth around it. While in viridans, there is growth. Okay. So it is resistant to optogen. And the other difference is that viridans is insoluble in bile, while pneumonia is bile soluble. So how I think of viridans, is the alpha here, has a lot of muscles, strong, nothing can stop him. Okay, so not even optogen nor bile. Tamam? Now, clinically, how would it present to you? So if a child comes to you complaining of otitis media or meningitis or even signs of upper respiratory tract infection, تمام, like pneumonia طبعا, okay, this will indicate, okay, this might be strep pneumo. So you will do these different tests to make sure that it's actually strep pneumonia. In viridans is usually associated with abscess, like brain abscess, liver abscess, subacute bacterial endocarditis, even dental caries as well. تمام? So this is how it really works. All right. Moving on to beta hemolytic group. هلا you want to understand one thing إنه strep pyogens تمام is categorized under group A Streptococcus and Agalactia is categorized under group B Streptococcus تمام. To be able to differentiate between them, we use the bacitracin test, which is as well an antibiotic. طبعا strep pyogen is going to be sensitive to it. As you can see, the inhibitory zone here, there is no growth. تمام. So it's going to be susceptible. While group B Streptococcus, which is Agalactia, is going to be resistant. As you can see, there is growth. There is complete growth around the antibiotic disc. Tamam? So this is the first way how we differentiate between them. The second way is the CAM test. So CAM is actually a factor. I'm not sure for what, but it's a factor that Agalactia has. And how would you know if it's positive or not? You're going to see like an arrow right here. This arrow indicates that, okay, this is CAM positive. This is more likely to be group B Streptococcus, which is Agalactin. Like, how are they clinically different? Usually, Agalactia, as I believe, it's part of the flora of the vagina. So usually during vaginal delivery, okay, these newborns could you know, could get infected with uh, Strep agalactia. So when they get infected with this group B Streptococcus, tamam, uh, you don't want to know further details, but you just want to know that it's associated with newborns, especially those who are being delivered vaginally. Tamam. For group A Streptococcus, which is Strep pyogens, they're usually, like they would present to you with like acute, those who they would have acute tonsillitis, for example, those who have rheumatic fever, or even post-strep glomerulonephritis, which is a kidney disease after an infection from strep pyogens, for example. So this is how they really are clinically questioned. And yeah, so far, any questions or everything is clear?
Everything is clear, Ismail, but I have a question like regarding the OSPE in general, if you don't mind.
Sure.
So for these types of questions, would we be like seeing like an MCQ style questions, or like they just like short answer questions?
Most of OSPE is MCQ style questions. That's one thing for sure. But sometimes there are short answer questions in OSPE. So like, yani, if we go back, look, this is a short answer question in OSPE. This is actually from the 10th batch, the mom. They would tell you that, okay, name this agar, explain why it is selective differential. This is an example. Okay, if you just wait just furthermore, you'll see I've also added other questions exactly OSPE style.
Okay.
So what you want to be worried about is that you want to make sure that you know how to write the names of the test, because sometimes they might ask you. Okay. Uh, and yeah, but they wouldn't ask details. Yani, tada, it's OSPE, yani, OSPE most of the times is very direct. It's pure memorization. Okay. As you can tell, Yani. So inshallah, it's gonna be easy.
Okay. Thank you so much.
All right, no worries. But now we're done with gram-positive bacteria. Come on. So now we're gonna move to gram-negative bacteria. Um, so let's go. So before we dig any deeper, so can you remind me what does selective and differential agars mean?
So selective will allow only one species to grow, and differential will allow me to differentiate between the between Ares and Epidermidis.
Yeah, so differentiate between like different organisms, right? That's what it means. Okay,
Yes.
thank you so much. Thank you so much. So now this is that's a perfect answer. So in gram negative, we use a different agar called the McConkey agar. Now, this agar is purely selective for gram-negative species. How does it do that? It has very high bile salts and crystal violets that only gram-negative species are going to grow. Tamam? How do we differentiate between different gram-negative species here? Lactose fermentation. So those who ferment lactose are going to be pink in color. Those who do not ferment lactose are just going to remain colorless or yellow. So if you think about it, it's exactly the opposite of mannitol salt agar. Tamam? Mannitol, it goes from pink to yellow. Here, it goes from yellow to pink if they actually ferment lactose. Tamam? So this is a high yield as well. You might want to take note of this one. Tamam. So in gram negative, طبعا McConkey is the test that would tell me if it's fermenting lactose or not. And based on that, I can طبعا يعني differentiate between them. So those who ferment lactose, by the way, I've never seen a single question on fermentation speed, so I think you don't want to worry about it. But just in case, those who ferment very fast are Klebsiella, E. coli, and Enterobacter. Those who ferment slow are Citrobacter and Serratia. Tamam? Uh, those who do not ferment lactose, however, okay, on the other hand, you want to do further tests so you'll be able to categorize them. The very first test you want to do is oxidase test. Now, if it was oxidase positive, tamam, then it's most likely to be Pseudomonas aeruginosa. If it was negative, you want to do a further test. And the further test is going to be a TSI or SS agar, which is short for Shigella Salmonella agar. And on that agar, if there is hydrogen sulfide production, which is basically those black colonies that you will see on the agar, then this will indicate that this could be Salmonella or Proteus. But if there were no black colonies, then it's most likely to be Shigella. تمام؟ طبعا, of course, I'm going to explain all of these. So let's start with the lactose fermenters. There are many, but these are the very high yield ones, the E. coli and the Klebsiella. So they're both gram-negative rods. Now, the very important uh, differentiator between E. coli and Klebsiella are two things. Actually, three, okay? Number one is the way they appear on the McConkey agar. Look, E. coli appears to be just um, E. coli just appears to be simple pink colonies, like as if it just got stained. تمام? On Klebsiella, sorry, in Klebsiella, okay, they're gonna look like it's a bit mucoid, okay? I think you can tell the difference, okay? And this is something that differentiates between Klebsiella and E. coli. تمام? And this is very important for you guys to know. Like they might not ask you directly, is this mucoid or not? But they would give you the agar dish in an exam and tell you what's this. So now you want to know that if it's E. coli or Klebsiella. Second thing is that E. coli is urease negative and Klebsiella is urease positive. Now, why am I emphasizing on this one? Because sometimes in a question, they will say that, okay, it ferments lactose, it's gram-negative, it has pink colonies, tamam, it's urease positive. Hello? And they might put you a dish there, the agar dish. Now, sometimes you might not be able to figure it out. Maybe the image is not the best quality. So you might want to know that Klebsiella is positive for urease. Second thing you want to know is that E. coli is not capsulated, while Klebsiella is capsulated. And E. coli can move, Klebsiella are fixed, non-motile. Usually, E. coli questions are more targeting UTI infections. Klebsiella, most of the times it's pneumonia, and usually it's a hospital-acquired infection. I even see it sometimes in liver abscess questions. But so far, this is it for Klebsiella and E. coli. So is this clear? Okay, I'll move forward. Tamam. So this is E. coli and this is Klebsiella. Now, if I give you this in an exam, how can you actually tell the difference? How can you tell that this is Klebsiella and this is E. coli? So there are a couple of things you want to keep in mind. First of all, tamam, this is gram-negative because it's pink. This is number one. Number two is that look at E. coli. It looks that it's slightly smaller than Klebsiella. Why? Because E. coli have no capsule, while Klebsiella does. So this explains why Klebsiella is going to look just a bit thicker than E. coli. Second thing, look at E. coli. It's all over the place. It's even in clusters, right here, for example. So this is because they're motile. They move. Tamam. Klebsiella, no, they're fixed. Okay. And this is how I actually can tell the difference between E. coli and Klebsiella, under a microscope. But now, moving on to lactose non-fermenters. Yes, you would get questions on others, but the high yield ones, like they always show up, are Salmonella and Proteus. Now, they're both gram-negative bacilli. They're both yellow or colorless on McConkey because they do not use lactose. And they're both motile. But Proteus is highly motile to the point that you can even tell it's Proteus just from the way it appears on an agar dish. This is called the swarming effect. Tamam? This is very important. When you see this word swarming, like swarming on an agar, it's definitely Proteus. This is just something that you don't even want to sit and guess and think about. Tamam? Both of them, as I mentioned, are gonna have black colonies on Shigella, Salmonella agar. Okay, but urease is positive in Proteus and it's negative in Salmonella. So this is something that could help you differentiate besides how Proteus actually looks on an agar, since they're very similar with most of their characteristics. Okay. Salmonella usually accompanies questions on food poisoning, gastroenteritis, typhoid fever, mainly food poisoning. Proteus, um, very rare that they will give you a clinical question on it, but it's usually associated with wound infections and UTIs. But Proteus, swarming effect. Sometimes they're not going to give you their guard, they're just going to tell you swarming. Swarming, Proteus. Tamam? Okay, moving on to Pseudomonas aeruginosa. This is this is an this is I would say this usually comes to people who are immunocompromised. It's very difficult for you to get this infection unless you have a very weak immune system. Okay, so um, there are gram-negative rods. They do not ferment lactose. They're oxidase positive, as we mentioned. But what could tell you that this is actually Pseudomonas is that it's gonna form these. It's gonna have like this blue-greenish pigment on an agar. This is very indicative that this is Pseudomonas aeruginosa. Okay? Is everything clear so far?
Yes,
Correct.
all clear.
Thank you. So lastly, we have, by the way, this is the Shigella Salmonella agar, these black colonies, as you can see. Um, Enterococcus, I've never seen a single question on this, but just that that Enterococcus, they come from, your like intestines. Cool. We have two very famous organisms that would cause, healthcare associated infections. You have the Enterococcus faecalis and Enterococcus faecium, and both of them are bile salt soluble. Uh, but yeah, that's all about it. So now let's answer some questions before we move forward. So, which of the following plates represent an organism that causes acute tonsillitis? So, who would want to answer this question for me? Anyone here?
B.
B. Can you tell me why?
It all on this is beta hemolytic, right?
Like, first of all, when you get this question, it could be شوي tricky. تمام? So, for you to be able to answer it correctly, تمام, you need to, first of all, think about what organism are we talking about? تمام? So, when we say acute tonsillitis, what do you think I'm talking about? So, could it be staph or strep? Let's start this way. Or actually, let's think gram positive or gram negative.
Gram positive.
Okay, gram positive. So what are we going to exclude? We're going to exclude this one because this is Proteus. This is swarming agar. So I'm going to exclude this option. Tamam. So gram positive, like, uh, do you think this is, can you tell me what organism is growing
This is
here?
Candida.
Exactly. So it's fungi. So it's either B or C. So this is beta hemolysis. So this could be either Pygens or Agalactia. And of course, it's not going to be Agalactia. So strep Pygens could actually give me acute tonsillitis. Like, what is this? Can you tell me what's this?
This is
This could be staph epidermidis, right?
yeah.
So I don't think staph epidermidis is going to give you acute tonsillitis. Usually, staph epidermidis is associated with, when they insert foreign objects to your body. So, for example, when they put a catheter or when they do surgeries, that's when staph epidermidis will actually do an infection. So, acute tonsillitis is going to be beta hemolytic strep pyogenes. This was last year, by the way, just to let you know. Okay, is this clear? I'll move on to the next one. So what's the name of this test?
This is DNA.
DNAs. All right. And is it DNAs positive or negative?
This is positive.
Positive. Okay. And what could be the organism growing here?
I think Staph aureus.
Yep, it's gonna be Staph aureus. Okay. And on a DNA test, if there was no signs of like hemolysis here, and it was just pure, uh, like no signs of change, that's gonna be Staph epidermidis. All right, cool. Play. Um, a 10-year-old boy, previously healthy, presents to the ER with pruritus, fever, vomiting after eating a fast food meal a few hours ago. Specimen was grown in an agar seen below. What's the name of this agar?
Salmonella Shigella.
Yes, this is Salmonella Shigella agar. And what causes the formation of these black colonies?
Ferrous sulfide reacting to form H2S.
Yep. So when you get this, honestly, when you get this question, just write H2S production. That's it. Simple as that. What is the most likely organism?
Salmonella.
Why not Proteus?
I guess because fast food as well.
Yep. So this is actually a case of food poisoning. Okay. So it's salmonella. Good job. Play. So now we are done with the bacterias, I think. Now let's move on to, minimum inhibitory concentration, Kirky-Bauer method. Now I heard that you know, you guys wanted me to yeah, explain this in details, so I will do that. So let's start with Kirky-Bauer method. Now, what are we doing here? So we have a petri dish, okay? Tamam, this is a petri dish, and these are antibiotic discs. Hello? Now, we are going to, of course, infuse the agar with certain bacteria, all right? And we have these three different antibiotics, and we're going to allow them to grow for 24 hours on the agar. Now, if there was an antibiotic disc with full growth, this is, by the way, bacterial growth. If there was full growth around it, then this means that the bacteria is resistant to the antibiotic. All right? But if there was no growth, and this is something we call the zone of inhibition or inhibitory zone, tamam, this means that the bacteria is susceptible to the antibiotic. And of course, we have levels of susceptibility. It could be intermediate or it could be susceptible. Tamam? And this really depends on the size of the inhibitory zone. And of course, the larger was the zone, okay, the more effective is the antibiotic. Tamam? So, yeah. And if they ask you in the exam, why is Kirky-Bauer method used? It's because it's very cheap and it's efficient and convenient. Tamam? And yeah. So as I told you, we have susceptible, intermediate, resistant. I don't think they're gonna ask about this because you have to know Kirky-Bauer method. Like they might ask you, they might give you this dish, okay, and give you like a few sentences about it and tell you what's the name of this method. So it's Kirky-Bauer method, and it's a qualitative method, okay? Qualitative. Now we move to quantitative, which is the minimum inhibitory concentration. Why do we prefer to use this one over the Kirky? Is because this one is very accurate, tamam, very precise. It tells you like exact concentrations of stuff, tamam. So when we say minimum inhibitory concentration, this is the lowest concentration of antibiotic needed to inhibit growth of bacteria. Tamam? So let's take a look at this illustration here. So this is, a test sample of certain antibiotic. Tamam? And here, as you can see, with this concentration, there is still bacterial growth. There is growth. But as you can see, the growth starts to decrease, right? Now, when you see that there is no growth at all, this will be your minimum inhibitory concentration. Tamam? So if they get you a figure in OSPE that there is growth on all of these colonies, but the first colony where you see no sign of growth, that will be the minimum inhibitory concentration. So let's, for example, take a look at this one. There's growth here, same as well. There's growth here. Even if there's one colony growing, that's still growth. But the very first one, the very first dish with no growth is the one with concentration of 8. So this will be your minimum inhibitory concentration. Is that clear? All right. So then there's another test that we do. It's called the E-test. Now, the E-test is basically, so we have this strip. It's an antimicrobial gradient, طيب, that's used to quantitatively determine susceptibility of microorganisms. And how would you know what is the minimum inhibitory concentration here? You're going to look at the last digit, okay, that's at the end of it right here. So it's going to be 4, tamam? So 4 is going to be your minimum inhibitory concentration. This is how you read an E-test. Hello? This is very accurate, but it's very expensive. That's why we do not use it. I mean, we use it, but we don't use it, you know, every day or whenever we choose to, because this is a very, very expensive test. So this is one of its limitations. But this is a very high yield example. I think if you understand this, يعني خلاص, you mastered this concept. So we have a specific bacteria called E. coli. Hello? And we use an antibiotic called amoxicillin. And as the E-test results, it told us that the MIC is 4 micrograms per milliliter. Tamam? The breakpoint of susceptibility is less than or equal to 8. So what does it mean when I say breakpoint of susceptibility?
I think when the breakpoint, so in general, MIC should be lower than the breakpoint of susceptibility in order to say that this antibiotic is susceptible to the bacteria. And in this case, like MIC is, I would say, is lower than the breakpoint. So that means that the antibiotic is susceptible.
So you're trying to tell me that if the MIC is lower than the breakpoint of susceptibility, the antibiotic is going to be effective on treating the bacteria. صح? That's what you said.
Yeah.
All right. Exactly. This is one part. The second part is that if it starts to get, okay, what if I told you that, that I'm using a concentration of 9, something more than 8. What does this mean to you? This means that, okay, the antibiotic is effective, but I'm requiring a higher concentration, higher than the breakpoint of susceptibility. So this means that the bacteria is starting to develop resistance. So if you use an antibiotic with a concentration above breakpoint of susceptibility, this means that this bacteria could start to develop resistance toward this antibiotic, because now you need to use higher and higher concentrations. Is that clear to you guys? This is very important.
Can you repeat it, please?
Okay, so our friend here, Yaqub, said that MIC طيب, should be lower than the breakpoint of susceptibility. And yes, this will indicate that the antibiotic is effective in treating E. coli. But here we said that the breakpoint of susceptibility is equal or less than 8. Tamam? So what if I told you that I'm using a concentration of 10 to treat E. coli? يعني the same concentration, مثلا I used to use 5 مثلا, the concentration of 5 microgram per milliliter of amoxicillin to treat E. coli. To kill the growth, okay, of bacteria on that petri dish. Now I'm using 10, for example, or even 12. This means that it's higher than the breakpoint of susceptibility. شنو the breakpoint of susceptibility؟ يعني if I use concentration more than 8, this means that the bacteria is starting to develop resistance. لأنه if you start to use very high concentration of something to kill concentrations that used to kill at very low concentrations before, this means no, there is some kind of resistance being developed. Did you get it? Well, لا.
So basically, Ismail, what you're saying is like if we are using a concentration that is higher than the breakpoint itself, then there's a possibility that the antibiotic is developing resistance.
The bacteria. Yeah.
Sorry,
Yes.
the bacteria is developing resistance.
Yes, طبعا. Because that's when يعني at that concentration it will tell you, مثلا if I'm using a concentration of 7, this is completely fine. Anything between 4 and 8, طيب, I'm killing the bacteria. It's all good. Even above 8, I'm still killing the bacteria. But if I'm using concentration, okay, more than 8 to kill these colonies, okay, this means that this bacteria is starting to develop what resistance. يعني أنا لازم أحط زيادة أضعاف الأنتيبيوتيك، ضيف أكثر وأكثر to make sure إنه all is killed. This is a sign of resistance. Okay? All good?
Yes, clear. Thank you.
Okay, alhamdulillah. طيب. So, the takeaway, the higher is the MIC, the less sensitive is the bacteria. And so, the more likely it is to be resistant. طبعا I flipped it. Why? لأنه people think the higher the MIC, the better it is, which is wrong. Always think about it. Imagine that you're getting money, okay, with the least the least input you could ever do. Isn't that so good? Isn't that what we all want? Instead of like putting on a lot of effort and a lot of work to gain that same amount of money. So same goes here. If the MIC was low, tamam, this means that if the MIC is low and I can kill the same bacteria with a very low MIC, that's very good. Means the bacteria is sensitive to the MIC, is sensitive to the antibiotic. Okay, this means it's susceptible. But if now I'm using very high levels of MIC, very high levels of the antibiotic, that's like, okay, which means that the MIC is very high now. This means that the bacteria is less sensitive, hello, to the drug, which makes it to be more resistant. So it's the same idea of breakpoint of susceptibility and that. So, but this is the takeaway, tamam, and I hope it's clear to you guys. Inshallah. So let's do some questions before we proceed to parasites, I think. So which line has the highest concentration of antibodies? And I want you to tell me why. Okay, who's gonna answer? I don't know.
To be
Yeah.
completely honest, I'm not sure if I understand the whole concept, but I would say the third line.
Okay.
Third line.
It is the third line.
I
But
really
I'll tell you.
I
Yeah.
really don't know why.
See why? I'm gonna tell you why. First of all, okay, you know, okay, I don't know how to shape this to you. So the highest concentration, okay, so it's the third line. Why? لأنه it's the one that that has like most of the bars being purple. طيب, because you want to keep trying to put these high concentrations of antibodies to kill the bacteria. تمام? Okay, till it's yeah, somebody raised their hand. Yeah, go ahead, ask. Hello. So is it like chat box or well, I'm not sure. Oh,
Someone said that shouldn't be a second line.
okay. Like, let me explain. What did I just say earlier? And at first, let's navigate this one step by step. Tamam. If I put tamam, uh, first of all, this is gonna be where there's no sign of growth. صح? This is where there's no sign of growth, right? Okay. So now when I keep trying to, now it's the only one that took so many distillation for it to actually die. Why is this so? Is because I keep adding these concentrations of antibodies. I keep adding more antibodies. It's not dying. It's not dying. Okay. So I added a lot of antibodies. Okay. Till it's oh my God. Antibiotics, not antibodies. Sorry. طيب. It actually works both ways. Okay. So this is why تمام I keep adding more and more for it to actually have no signs of growth, for it to be clear. تمام. Do you get the idea? In second line, I just added a few. خلاص, there's no more growth. You get what I mean? So the more tubes you see, the more you're adding. So the higher the concentration is going to be. You get what I mean? Is this clear? Well, you guys didn't get it.
Honestly, I didn't. I didn't get it.
Like, let's go back. Let me just go back. Like, uh, look at this. I started with concentration of 0.5. تمام? And then I added concentration 1, then 2, then 4, then 8. So I kept adding these concentrations till I reached 8, till I made sure that everything is gone. There's no sign of bacterial growth. So the more, يعني, the more tubes I utilize, the more concentration I keep adding. I keep increasing the concentration, تمام, till I make sure that there is no signs of growth. Did you get it? ولا لا?
Yeah, it makes sense now.
Yeah, okay. Uh, the girl who raised her hand earlier, did you also get it? Well,
Doctor, in the question you just showed right now, a few minutes ago, the one with the 4 lines and which one had the highest concentration? Yeah, here.
yeah.
The second line is most effective against the pathogen, right? Because the least
See,
concentration is required.
yeah, if you think about it that way, okay, that's a very good way to think about it. Yes, tamam. But again, yes, you're right. Okay. But the one, just to make sure that the others are not confused, the one with the highest concentration is going to be C. So did you get why? Because I kept adding and adding and adding till I see no sign of growth. Hello? You
So the
got it?
more antibodies there are, the more resistant.
Antibiotics.
Oh.
Oh yeah. So if you want to think about it, I'm sorry, it's a typo. The more you add, okay. So I cannot really tell because I don't know if they're using the same organism. But let's assume that they're using the same antibiotic, same organism. If you keep adding and adding and adding, okay, like this one means it took a lot. So this could indicate resistance. You cannot conclude from this image only. You have to do different tests, okay? Like you need to do the E-test, you need to find out the breakpoint of susceptibility. Okay, so from the breakpoint of susceptibility, you can tell if there's any resistance or not. Because as I mentioned earlier, so let's say the breakpoint of susceptibility is 8 or less. Tamam? So if I'm using a concentration of 10 or 12 or 24, this could indicate that my antibiotic is not being as effective as it used to be before on E. coli. Okay, so this is how we really assess susceptibility. I know it might be confusing, but you just need to just think about it for a minute. Okay, and I hope it's clear. So is everything clear? Shall we move on? Well, do I need to repeat it? I'm happy to repeat.
Alhamdulillah, it's clear. Thank you, Doctor.
Okay, you're welcome. But I hope things will also be clear after this question. Like, which of the following graphs shows the most effective concentration of the drug on killing the microorganism?
The one in the middle,
Why?
because the MIC is much lower than the breakpoint.
Okay.
If it's higher than the breakpoint, it would it would say that it's resistant, right?
Okay, so yeah, so C is definitely wrong. But my point, for example, let's say I'm confused between A and B. Why would you say that the answer is B? يعني صح, C we're excluding because MIC should be lower than the breakpoint. That's correct. But it's asking most effective concentration on the drug, of the drug on killing the microorganism. So what do you think?
So basically it is between, as you said, like it is between the first and the second one. But since it is asking for the most effective concentration, it would be A, since it is more susceptible to kill the microorganism. But
Shouldn't it be B
uh,
because the MIC is lower?
yeah, that's the point. So it is if again, remember, as I told you, the higher the MIC, the more resistant it would be. Tamam? So the lower the MIC, the more effective the drug would be on killing the microorganism. Because the microorganism itself is sensitive to the drug. You get what I mean? شوف, this is high, a bit higher than this one. So I would always prefer to go with the lower MIC. More I prefer. This is the science behind it. Did you get it, or you have questions in your mind?
yeah, it makes sense now. Yeah.
Like, alhamdulillah, thank God. Okay. This is a question from the past. Fewer questions. All of them were from the past, by the way. So yeah, let's do this one. A microbiologist was measuring the diameters around each zone. What is the name of these zones? Yeah, anyone?
Inhibitory zones,
Yep, inhibitory zone. Correct. What's the name of this method?
Kirky or Kirby Bauer.
Yeah, Kirky-Bauer. Okay. Now, do you think this is true? The larger the diameter, the more effective is the drug?
No, not really.
Why?
Because it should be the more susceptible. Or
I don't get you.
basically, how can I say it? The bacteria is more susceptible to the drug.
So, you're, can you rephrase it? Shani, I need to make sure I understand you clearly. Let's say, I use a specific concentration. So, for example, this one, okay, and it had to kill all of these. So, don't you think the larger the diameter, the more effective is the drug? Like it's killing more.
In some way, it's not true.
I mean, I think that the larger the diameter, the more effective is gonna be the drug.
Uh,
Unless if I'm wrong,
so
please correct me.
so basically, I think I have encountered this like question before, and I immediately said yes, but the answer was actually no. And the reason why is because in general, like when we look at the diameter, not only you would look at the diameter and say, oh, well, this is the larger the diameter, the most effective. You need to compare the diameter with the standardized value, and it should be larger than the standardized value. Because if it is smaller than the standardized value, I think it is like there is a specific table that shows all the standardized values. So you compare the
Yeah.
with the standardized value, and it should be larger. So looking at the diameter itself and say, oh, it is more effective, that by itself is not enough.
Yep. All right. Thank you. Thank you, Jacob. All right, so we're gonna move on. I think there's one more question. Play. So this one. This is, you know what's this called?
E-test.
All right, and can you tell me like an advantage and a disadvantage for it?
It's really effective.
Okay.
One disadvantage is that it's expensive.
Yep. Play. Thank you. So do you guys want to take a break or shall we keep going?
1 minute break.
Okay, sure. Let's do that. Let's have a 1 minute break. Okay. So we're gonna now do parasites. It's gonna be way long. Uh most of it is any memorizing honestly, but I'll try my very best to make it easy for you. So the way I divided the parasites, we have GI parasites, we have blood parasites, and we have tissue parasites. Okay, so these are different locations where they could be, and I'm gonna start with the GI. Like for GI, this was not in your lab. I don't know why, although it was in the OSPEs of previous years, so I just happened to include it. It's called Giardia intestinalis. تمام. Giardia intestinalis. Now, very high yield. You need to know its fecal oral route. Yani, it's very important. Okay. And it does not cause any extra intestinal diseases. It's just located in the intestines. The only thing it's going to do is going to cause inflammation of the colon and it's going to cause malabsorption. Oh, sorry, not just the colon, the entire intestines. Okay. It's gonna cause inflammation leading to malabsorption. Pardon? And if you want to order a sample for a guy you think has Giardia intestinalis, you want to order stool sample. Tamam? How do you see it? So, you'll just see like the cyst and trophozoite, like kind of like Loki have eyes and a face. So this is how I really memorized it. All right, this is a cyst and this is a trophozoite. Trophozoite is usually the diagnostic stage, and the infective stage would be the cyst. Hello? Is everything clear? Inshallah? But I'll move on to the other one. So, E. histolytica or Entamoeba histolytica. Okay, so we have different species of Entamoeba that would cause amoebiasis, but the one that is clinically significant is E. histolytica. Okay, and the way I think about E. histolytica, like Entamoeba eats erythrocytes. EEE. Entamoeba eats erythrocytes, which are the red blood cells. طيب, keep this in mind for later. I'm gonna show you why. So again, fecal oral route, anal oral route, same thing. Okay. The cyst has four nuclei, and this is very, very important when you come to diagnose because they could give you a cyst with two nuclei, eight nuclei, four nuclei is indicative that this is E. histolytica. Okay. And since it's fecal oral route, you want to order a stool analysis, tamam, and you will have a cyst and you'll have a trophozoite. If you look at the trophozoite, it's like a huge cell and there are like multiple cells inside. These are the erythrocytes that the E. histolytica eats. So these are the red blood cells right here. So this is how I remember it. Okay. E. Entamoeba eats erythrocytes. Play. Cyst, again, is going to be the infective stage, and trophozoite is going to be the diagnostic stage. Tamam? I hope this is very clear. This is very important. Tara. It causes extra-intestinal diseases. Yani it can go to the liver, tamam, and it can form, like abscess there. It could go to the lungs and even cause pneumonia or whatever. It could even migrate all the way up to the brain. طيب? So it's very important. We use a thing called iodine mount. طيب? And this is how it's gonna look. So see, this is another another species of entamoeba called Entamoeba coli. This is not E. coli. This is Entamoeba coli. Okay. And as you can see, it has more than 4 nuclei. So this is a way that you can differentiate between E. histolytica and Entamoeba coli. Also, if you can appreciate the difference between the Entamoeba coli trophozoite and the E. histolytica trophozoite, it just has like one cell. Okay, even the color, the texture looks different. طيب? So it's worth noting that. Then we have the wet mount. طيب? We usually get these samples from watery stools. And look at this. This is a trophozoite of an E. histolytica. Okay. And how can you tell that it's E. histolytica? It has so many red blood cells inside the trophozoite. Like it's packed. Okay. So this is something that you also want to know. And usually they're irregularly shaped. Tamam? Now, this is quite important. This is an ulcer, okay, in the colon epithelium, okay, and it's a flask-shaped ulcer. This is high yield to know flask-shaped ulcer. Okay, this is how it will leave the intestine and cause extra intestinal manifestations, the ones that we talked about here. Okay, so this is how it does that. So it's very high yield. So we're done with E. histolytica. We're done with Giardia intestinalis. Now we move to Plasmodium, blood. So Plasmodium, aka malaria. This is what you want to know. طيب. I'm not gonna go through the entire thing, but you might want to know the vector and the definitive host for Plasmodium is the female Anopheles mosquito. If you write mosquito in the exam, that's not gonna be an acceptable answer. You're gonna get 0 for that. You have to clearly write female Anopheles mosquito. طيب, because a male Anopheles does not work. Just mosquito alone does not work. It has to be female Anopheles mosquito. تمام? Okay, so how does it start? So there's this mosquito, طيب, a female Anopheles mosquito, that's going to inject sporozoites while it's just sucking blood. Okay, so it's gonna inject the sporozoites, and they're just gonna, go to the liver and then infect a liver cell that will give us a schizont. All right, and then this schizont is gonna keep becoming larger and larger and larger till it ruptures, and it's gonna release things called merozoites, which are going to infect the red blood cell, and then it's gonna form trophozoites within the red blood cells. Okay, so yeah, that's all the high yield you want to know from, practical point of view. Infective stage is going to be the sporozoite. Diagnostic stage, you can have a schizont, you can have a trophozoite, you can have a gametocyte as well, because these trophozoites, tamam, they can either continue to be mature trophozoites and then become schizonts again and repeat the entire cycle, or they will differentiate and become gametocytes. Hello? Gametocytes are usually like banana shaped. I'm going to show you in a bit. But there are multiple species of Plasmodium, but the one that we are very concerned about is falciparum. It's the one that causes the very severe infections in humans. And a clinically relevant concept for P. vivax and P. ovale is that they have a latent form, like a hypnozyte that's in the liver. Okay, which means that they stay latent if something happens that would stress them out, they'll be reactivated again. Okay, so it's a bit tricky when it comes to Vivax and Oval. But when we come to collect samples, of course, it's blood. We have two ways. We have a thick film and we have a thin film. Thick film is going to destroy the blood cells and it's just going to expose for me the, the malarial parasites, just for me to see it properly. The thin film, no, red blood cells are preserved and you can actually see the trophozoites clearly. I'm going to show you. So this is a thick film. So all the blood cells here are destroyed and you can see these dots. These are chromatin dots, okay, of the malarial parasite. Tamam? On a thin film, this is a red blood cell, and you can see like a ring-like structure. These are the trophozoites within the red blood cell. And these dots are the chromatin dots that you just viewed right here. Okay? So we have a thick film and a thin film. Tamam? I hope it's clear. And these are the gametocytes, by the way, that I just talked about. They're banana-shaped, and yeah. So we're done with Plasmodium. Is that clear to you guys, or is there anything that I need to repeat? All right. So moving on to Leishmania. Leishmania, I don't know why, but like people usually ask a lot of questions from Leishmania. Like I've seen like 3 years in a row, and there's a lot of questions from Leishmania. So it's pretty high yield. So Leishmania, it's an indirect life cycle. Okay. طيب. It starts, within a sandfly, and then when the sandfly goes and, try to suck blood again, that's where it's gonna infect the human being. Okay. And this is when these cells are going to, are gonna be phagocytosed by neutrophils, tamam, and then they're gonna form promastigotes. Hello? And then these promastigotes are gonna then become amastigotes. طيب. The promastigote is the infective stage. And how do you know it's the promastigote? It has a flagella. Like, the sperms, how they have like these flagellas, how they swim. Same thing with promastigotes. And before diagnostic stage is going to be amastigote. So it's going to be this large macrophage with, dots and dots and dots of cells that have been, which were the promastigotes that became amastigotes and they were like phagocytosed. So let me show you. So this is the promastigote. As you can see, it has flagella. طيب. It's actually a bit similar to Giardia intestinalis, right? The trophozoite there. But what makes it different that these flagellas are really big and this one does not have like a face-like structure or like an actual face when you stare into it, Like look, this this is a bit creepy, you know. This is just different. So this is a promastigote. It's very important, guys. Okay. A mastigote. So this is a macrophage. And as you can see, dots, dots, dots. It's like glitter, right? Okay. And yeah. So this is infective. This is diagnostic. But we have different kinds of Leishmania. طيب. We have Donovani, we have tropical, we have Mexicana, and we have Brazilian. طيب. The visceral Leishmania is the Leishmania donovani. Okay, it usually targets the reticuloendothelial system, which is the liver, spleen, and bone marrow. So if you suspect that this Leishmania is Leishmania donovani, you want to take sample from from bone marrow. It's the easiest to do. Hello? Usually they come with bleeding, jaundice and all that. So Donovani, Leishmania donovani. Cutaneous, like skins, like this one, for example, right here, we have both, two, tropical and Mexican. Just shortcutted it into Tropica Mexicana, okay? So, but this is not the actual name. It's tropical and Mexican, okay? So, for cutaneous, it's the Tropica Mexicana. And this is how they form these cutaneous skin lesions. And for a sample, you want to take a skin sore, okay? And that's the thing that you're gonna analyze. Mucocutaneous, okay, it's similar to cutaneous, but the difference is it involves even deeper layers. Okay, and it's actually gonna be a necrotic ulcer. Okay, so this is how you can tell the difference between cutaneous and mucocutaneous. Mucocutaneous is like actively bleeding, as you can see. Here it's just simple. But again, you would do a skin sore. Okay, I hope this is clear. So I'm gonna move on. Then we have Trypanosoma cruzi. But Trypanosoma cruzi. So what's the organism here? It's gonna be a bug, which is the triatomine bug. Okay, so this bug, tamam, is gonna go, okay, and take a blood meal. طيب. This blood meal, طبعا, you need to keep in mind that that usually it's it comes from like feces of some organisms that actually are reservoir for Trypanosoma cruzi, like dogs, rats, and whatsoever. Hello? Then this this bug is gonna give in the metacyclic triple mastigote, hello, and it's gonna fuse itself within like multiple layers of an infected tissue, and it's gonna look like it's like a sandwich, okay, of amastigotes inside cells. This is very distinctive of Trypanosoma cruzi, and this is how it looks. Look, this is very important. تمام. طيب, this is the metacyclic triple mastigote. How would I say it's metacyclic triple mastigote is because the way it abnormally looks. Usually all the mastigotes that we've been through, they're like large ones, right, with flagella and all. This one is very irregularly shaped, okay, because it's metacyclic. So metacyclic triple mastigote is the infective stage. And for the diagnostic stage, it's gonna be either triple mastigote or tissue amastigote. And so far, all the questions I've seen in the past were tissue amastigote. And the sample you're gonna take is a blood sample or the infected tissue biopsy. And when you do biopsy, you're gonna look at this. Tamam? Is this clear to you guys? So the bug is very important. Hello? طيب. Now, طبعا we have Trypanosoma cruzi, we have Trypanosoma brucei as well. طيب. The cruzi one is gonna be clinically associated with a Romanian sign, okay, as you can see right here, and they would have this kind of skin lesion called a shagoma. طيب. So these are diagnostic for Trypanosoma cruzi. Amma, we have this thing called the Trypanosoma brucei. This one, or is called the sleeping sickness. Tamam. Uh, the the vector will be the tsetse flies. All right, and we have two. We have T. brucei gambinese, and we have the rhodocines. This is in the west. This is in the east. طيب. Now, what you want to know? Look at the trypanomastocysts. How different they look from the ones here. This actually you would see like they have like an eye, like they're like a kind of worm or something. Okay, so this is how I really differentiate between them. Cool. And they have so many slopes and they're very irregularly shaped. Okay. Diagnostic stage will be the triple mastigote, and I would take a blood sample, I would take CSF sample, or even like a lymph node aspirate. Like, so you have three different samples that you can collect. And another question that might show up as an MCQ, I believe, would be like, what would be the reservoir for gambinese? It would be humans. But for the other one, it's going to be animals. طيب. So, Trypanosoma, we have two, cruzi and brucei. طيب. Cruzi, hello, as we mentioned here, they will have amastigotes within the infected tissue. This is very diagnostic for them. طيب. And they would come with Romanian sign and shagoma. While the brucei one is usually called the sleeping sickness, and they would come with different shapes of metacyclic trypanomastocyte, as you can see here. طيب. And that's all the high yield that you need to recall. And of course, the name of the bug. And look, this is how they would look. Since there's this chancre, okay, right here, the sign of chancre. This is a child who has an advanced disease with CNS involvement. You can look that he's disoriented and stuff. So usually the one with Brucey is more severe because it also involves CNS involvement. Tamam? Okay. Toxoplasma gondii, uh, or the cat disease, how people call it. So the definitive host would be the domestic cat. طيب. This is an obligate intracellular parasite. Why is this clinically important to know?
because it can't like survive that much outside organism.
Yeah, this is one thing. Yes, it's an obligate intracellular parasite. So it's one thing. It cannot survive outside an organism. Plus, it's very difficult to treat because it has to be intracellular, okay, inside a cell. And as you can see here, this is a tissue cyst, طيب, and uh, this is very, very, very diagnostic of Toxoplasma gondii. This is the infective stage. This is how it's going to look. And then we have tachyzoids. But let me explain to you something. So if it comes from undercooked meat, it's going to be a cyst. But if you see in a question, oocyst, then it came from cat feces. Hello? So this is very important for you to understand. And when they're being ingested by a macrophage, this is when they become a tachyzoid. Okay? As you can see right here. So these are tachyzoids. And when these cysts are trapped within a tissue, then these will be called bradyzoid, slow growing, slow moving. Tachyzoids are very fast moving. Okay? So this is the main difference. So Toxoplasma gondii, infective stage would be tissue cyst. The diagnostic stage would be a tachyzoid, as you can see right here. So yeah, I hope it's clear. Then we have Babesia. Now, Babesia, this is the tick disease. Okay, it's a zoonotic infection, and humans happen to be opportunistic hosts. Okay, so this will happen to those who are very immunocompromised. This is when they will get, such manifestations. Now, how it all starts, the tick itself is going to become infected. It's going to have these sporozoites. And then when they go for a blood meal, they're going to inject these sporozoites into the blood. Tamam? And those sporozoites are then going to become trophozoites, as you can see here. And then these trophozoites are going to become merozoite. And this is very diagnostic for Babesia. Okay? So, infective stage would be sporozoid. As you can see here, this is just simple sporozoid. طيب. This one, okay, is a merozoite. طيب. It's very important to notice the merozoite. This is very diagnostic, and they always usually get this one. Okay, most of the times. I'm sorry, by the way, this is the trophozoite. This is the ring structure that you see. Okay. So yeah. And the sample we collect is blood because this is mainly in the blood. And one other thing that this is usually transmitted from human to human via blood transfusion. Okay, Like, we're done with the parasites. Do you guys have any confusion from all the things that I've said so far? Like, do you want me to repeat any of the parasites that we talked about? Well, is everything clear?
Everything is clear. Thank you.
Okay, right. Then most of this is pure memorization. Okay, so good luck with that. So now we have helminths. طيب. In helminths, the way they're divided is, يعني, you have these simple protozoas that we just discussed. Sorry, there's not the helminths, but the parasites in general. They're divided to protozoa and helminths. Helminths are the worms, tamam. And the way they're divided, we have flatworms and we have roundworms. Hello? So this is how, this is what we're gonna basically be discussing. Most of these right here. So let's start with Ascaris lumbricoides. So this is mainly from soil. It's a soil transmitted roundworm. Okay, as you can see right here. Tamam. What happens is that this roundworm طيب, enters ماشي, via they would start, first of all, they're in the soil, تمام, and they will have these embryonated eggs that they're released, تمام. These eggs will enter, طيب, to the human body, حلو, and then, ماشي, they will go all the way to مثلا the intestines, the lungs, wherever they feel good like going, and then they will hatch there, and they will release the worms, تمام. So these are the ones that you must know. This is an unfertilized egg, okay? Uh, and this is a fertilized egg. This is the one that actually gets ingested, by the way. This is an embryonated egg. طيب. If you can just take a look, it's the way it looks, it's very like has very dark contrast around it. And you can actually see that it has something inside. So it's embryonated. And this is an egg that contains a larva, okay? Yeah. And then the worms are going to be released inside, like the egg hatches inside. So diagnostic stage, okay, you would see either of these eggs, plus you're going to see the worm itself. طيب. So look at this. This is a liver, okay? We're in the liver here, and this is a worm right there. And this is the intestine. This is diagnostic, okay? And it's very alarming. So yeah. Moving on to Trichuris, Trichuria, or the whipworm. So again, this is a soil-transmitted roundworm, okay? Fecal oral transmission again. Hello. The way this happens, again, we have this embryonated egg that gets ingested. And when it gets ingested, this is when the larvae is going to hatch in the small intestines. And usually, okay, upon endoscopy, you're going to see them like uh, hinged to a wall right here, or right like to the intestinal wall. Hello. Now, the embryonated egg, the infective stage, okay, what makes it different and easy to tell is the terminal plugs they have on the sides. And honestly, if you look at the middle, like it's very glittery, it's like a galaxy, you know what I'm saying? And these are like the terminal plugs right here. So this is very, very specific to whipworm. Okay. Usually, those people present with anemia, okay, due to some bleeding and rectal prolapse. So, if they get you a scenario that there's this child who is pallor, okay, and has been feeling very extremely fatigued, and he's having all signs of anemia, all right, and upon physical examination the child tends to have rectal prolapse. Like when he sneezes, okay, you'd actually see that the rectum is, being, you know, outside and outside of its place. So this is rectal prolapse, and this is seen in whipworm. So it's very important to know how, they would present. Hello? And yeah, pinworm. Pinworm, this is very commonly seen in children. Okay, so it's a fecal oral transmission, and it's usually self-transmitted. Like the child would be itching the perianal area, okay, and when they itch, like their child, so they would just take their hands and put it in their mouth, right? So this is how it's gonna be transmitted again and again. They keep doing it to themselves. So these eggs are gonna be on the perineal folds, ماشي. There'll be, of course, embryonated eggs that are going to be ingested by a human. Hello? This embryonated egg is the infective stage, طبعا. And then when it gets in, all the way to the intestines, all right, these eggs, yani, could then hatch, give us a larva, or could even give us like a full worm. And the diagnostic stage, of course, it's a child. When you would do something called as a Scotch tape preparation. This is very important. This very high yield, like high chances it might show up on the exam. All right. When you do a Scotch tape preparation, which is this process right here, you're going to see that there are eggs on the perianal folds, and this is diagnostic when you find those eggs on the perianal fold. So, infective stage is the embryonated egg, and the diagnostic stage are the eggs that you're going to find around the anus. All right. And how do you do that? You will do the Scotch tape preparation. It's very important. And طبعا, the embryonated eggs, honestly, the way I memorized it is just it has a pink contrast. Of course, not this is not the best way, but there's so many embryonated eggs that you want to know. So this is how I memorized it. Anyhow, moving forward, hookworm. So hookworms, طيب, again, infested soil. All right. What happens usually, there's eggs in the feces. Okay. These eggs are gonna hatch and give you this rhabditiform larvae. And then there's this larvae that's called the filariform that's going to penetrate the skin and it enters. So it enters as a larva itself. All right. That is infective stage. So the filariform larva is the infective stage of a hookworm. But when you see eggs in a stool analysis, that is diagnostic. So usually these kids, when they go to rivers and swim, okay, this is how these larvae would penetrate the skin. Okay. Now, its ability to penetrate the skin is because of certain enzymes. It has to be able to break through these, you know, multiple dermatological barriers. So they have protease and they have hyaluronidase. Okay. Or two species that causes this manifestations. Ancylostoma duodenal and Nicator americanus. It's very important, guys. They present with iron deficiency. It's high yield. Iron deficiency, hookworm, filariform larva. Okay. This is the diagnostic stage. This is the egg. I don't know if you can like take it and like notice this egg seems to be like clusters, like it's squished, you know what I mean? So this is how I really memorized it. And this is the filariform larva. Okay, this is infective and this is diagnostic. Is this clear? This is very important. All right. So, tiniasis. Now, tiniasis, humans are the definitive host, Pelu, and we have multiple species. We have Solium, we have Siginata, okay, and this basically is because they have different reservoir hosts. Like they it can either come from a cattle or come from a pig. Cool. What happens is that there's for example in undercooked meat, okay, there will still be this larval form, okay, within the meat itself that's gonna be infected, sorry, that's gonna be ingested by a human, okay, and then this meat, mashi, will have these larval forms that then through stool analysis, you're going to see that there are actually eggs or gravid proglottids in human feces, as you can see right here. These are very indicative of tiniasis. It has very thick outline, as you can see. Tamam. So, yeah. طبعا, we have, as I told you, T. solium and T. saginata. T. solium comes from pigs. Saginata comes from cattle. Now, solium is the one that causes very severe manifestations because when the eggs are ingested, they would do this thing called the cyst cirrhosis, okay, where they will cause a seizure. But how would you know that this is T. solium and this is T. saginata, since the eggs look similar in stool analysis and that? The only way you can tell its difference is from the larva form. Look, this is the larva form of the T. solium. It has this thing on top, okay? Like you remember, like when you like see a flower, like this thing in the middle, it's very similar to it. Okay, it's called the rostellum. T. solium has it. Saginata, it's absent. Even look at the suckers, the way they're like here, it's like I think it's more than four. Here it's just like organized in this fashion. You know what I mean? But anyways, the rostellum is the one that can tell you the difference between them. Hello? I hope it's clear. Then we have the Enterococcus granulosus. Now, this is the infection here is accidental, okay, and even the patient himself will not be able to know that they have the infection unless they, for example, do certain tests and it will be found incidentally. So let me tell you more about it. Actually, I saw like a real patient a few weeks ago who had this disease. So basically, it comes from dogs or sheeps. So usually those patients who have farms and stuff, okay, and play with these dogs and sheep could, be susceptible to be infected with Enterococcus granulosis. So what happens is that you will have, you will have these embryonated eggs in the feces of the dog, all right, that will be ingested by the child. And as I told you, the child, any, uh, I don't think that they're well hygienated, so they're just gonna, whatever is in their hands, they're gonna put in their mouth because they love to explore, right? So that these embryonated eggs are gonna be ingested, all right, by the child, and then they're going to form this thing called a hydatid cyst, okay, which is literally right here. So in the scenario, the patient that I saw, they were doing an X-ray, and the father found something in the lungs, meaning something very tiny, and thought it was abnormal. And then later, when they did further checkups and they even looked at the liver, they did ultrasounds, they found that there are hydatid cysts. Okay, so infective stage is the embryonated eggs. Okay, the diagnostic stage is the hydatid cyst. It could be in the liver, it could be in the lung. All right, so it could even go all the way to the brain. But yep, so a hydatid cyst is the diagnostic stage for Enterococcus granulosis. I hope this is very clear. Then we have the schistosoma. Schistosomas, um, so this is also high yield, okay? So usually patients, they're free swimming and all that. And then there will be this cercaria, okay, that's gonna actually penetrate the human skin, okay, right here. It comes from snails, by the way. So the host is a snail. Like you need to think of that one, freshwater snails. Because sometimes in OSPE, they will give you these pictures, all right? And then they will tell you which of the following has snail as a host. So you want to keep that in mind. Then they're going to form this cercaria, all right? And it's going to penetrate the skin. And then, all right, it's going to give us there are different kinds of schistosoma, all right, and depends on where they cause the infection. Okay, so we're gonna have different what to say, different kind of eggs that you're gonna see in the stool. All right. So look, not necessarily the stool, by the way. It depends where it's infecting. So we have the hematobium schistosoma, which affects the urinary system. All right, so you're gonna see this egg. How would you know that this is urinary schistosomiasis? From the terminal spine right here. This tiny little spine, this is called the terminal spine. And you'll collect sample from the urine. Amma, if it was uh if it was intestinal, it could be Mansoni or Japonica. How can you tell the difference? Look, this is the Mansoni egg, and they have a lateral spine. While here, they barely have a spine, so as you can see, it's just like fully round. This is very important. Okay. So again, terminal spine, if it was urinary, lateral for Mansoni, and like almost no spine for the Japonica. Okay. You get these from the stools, you get this from the urine sample. All right. I hope this is clear. So let's answer some questions before we proceed. So
Ismail,
at leaving, oh yeah, tell me.
sorry. So basically, I think you said like in the previous slide that sometimes they give you the life cycle and point
No,
out.
not the life cycle. They would tell you the host. Like they would tell you, oh, which of the following has snail as a host? You know what I mean? So that's when you need to be able to be familiar. Like they're not going to tell you schistosoma, they're not going to tell you anything. So these details are very important. So for any parasite, any helminth, you need to know three things. Do not enter the exam without knowing the infective, diagnostic stage, and the host, and as well the sample that you want to collect, you know, that you want to collect to see if they have the disease or not. These are very important. Okay? But let's solve some questions. So we have an anemic patient infected with an intestinal worm. Which of the following is the most likely the egg form of the worm, which will be found in a stool sample? What?
بس شوف it can be A and C عشان hookworm also has anemia and at the same time trachea also can be anemia.
All right. This look, it told you it's anemic. It's an anemic patient.
أيوة it could be both A and C. Also trachea can be anemia.
Sorry, I must have the last part. It could be A and C and
It could be either C and A.
okay. طيب. So what would you go with if you had these four options?
I would probably appeal it.
Okay. And I would go with A. Okay, so let's just go back real quick. طيب. In trichuria, you're gonna see this egg. تمام. ماشي. بس they didn't tell us they didn't give us طبعا more details مثلا if they had rectal prolapse or whatsoever. بس they clearly told us يعني they clearly told us شو they told us there is anemia تمام and stool sample. Wait, I can't find the egg. Or yeah, it's this one. Okay. So يعني is it this one? I'm not sure. Yeah, I think it's this egg. تمام. So yeah, uh, I would just go with whatever symptoms they gave me, and I would go with the answer. Try when you do OSPE questions, don't overthink the question. خلاص, you told you anemic, egg, stool. They didn't mention any other things. إذا قال لك rectal prolapse وقال لك هدول, okay, I would go with C. عرفت؟ But just to be on the safe side. Okay. طيب.
Regardless, this sounds like an appeal question.
All right. Okay. طيب, for B,
يعني
do you know what?
normally they would list all the يعني the physical signs of
لا
the
صراحة normally
Yes.
they will get you يعني شو I'm not يعني I don't know about the wording of the questions but most of the times يعني it's very clear, very straightforward. And remember, these questions are written by students. Like, this is not the exact and I copy pasted this question from the past batches. Uh, Honestly, I think it's gonna be clearer than this, inshallah. Or even sometimes they're gonna get you very, very weird written questions. But to be on the safe side, don't overthink it and go with the one that's like خلاص يعني, your most confident answer. You know what I mean? Or as your colleague said, you can appeal it. Like, what's B? B is for what? These eggs.
Enterobius.
What is it? Or must not?
Enterobius.
Okay. And D?
I think this is Ascaris. Oh,
Yep, this is
yeah.
Ascaris. This is Ascaris. Good job. Okay. And these are the terminal spines, by the way. Yeah, terminal spines. Okay. Play. Which one of the following parasitic stages shown in the attached is the infective stage of Leishmania donovai?
A.
Play. I want you to tell me what is B and C as well.
I think B are the gametocytes of
Gametocytes of the
Plasmodium.
P falciparum. All right. Play. C.
And C is the metacyclic trypanosomes of Trypanosoma cruzi.
Okay. Play. Good job. Nice. Wait, do you think it's Cruzy or Brucey?
I think this is cruzi.
Okay. Nice. So, is it clear for you guys? Well, we need to go back.
B is what?
B is the gametocytes of
Of
the P falciparum.
okay, thank you.
All right. Right. All right. Which one of the following represents Ascaris lumbricoids? Our friend طبعا did the job. It's D. Okay. Which one of the following has a snail as a host?
It should be none of them. It's none of the above.
Exactly. It should be none of them. Thank you very much. So, because none of them is schistosoma, okay? None of them will show you these different eggs, you know, this one and that one, and these. Like one does. All right. Which one of the following is diagnostic stage of Leishmania?
B.
B. And what's B?
Amastigote and macrophages.
Yep, exactly. All right. Thank you so much. Like, come on. Good luck. Now we're done with parasites. We're done with helminths. We're done with bacteria. Let's do fungi. Come on, fungi is very quick. Um, I don't like fungi. This we have to do it. So, fungi, you want to know that it's multinucleated and multicellular, except for yeasts. The yeasts are unicellular. About the structure, okay. So it's hyphae. So usually it's like a mold, all right, made of multiple hyphae, and multiple hyphae will give you this thing called mycelium. Okay. Fungi is not something that you will normally get. Okay. It's an opportunistic pathogen, meaning that if you have a chronic disease, if you have some immunocompromisation, that's when you will get it. Hello? They have the ability to produce asexually and sexually. All right. So you can also keep that in mind. Oh, yep. Let's start. So, how would you culture fungi in a lab? So, the best way, you have the SDA agar. All right. So the SDA agar is gonna give you this figuration of fungi. Okay, and this is usually like Candida albicans, but anyways, this is fungi on Sabarod dextrose agar. You have to know the full form. You have to write this out because sometimes they're not gonna accept short forms. So you have to know what's called Sabarod dextrose agar. Okay, and yeah, this is very important. So now, how do we look at fungi microscopically? So we can either treat them with potassium hydroxide if they were dermatophytes, like if they were fungi that causes like skin manifestations. If they were dermatophytes, you would treat them with potassium hydroxide. If it was candida, you're gonna just give it normal grab stain. Okay, and this is how it's supposed to look. Hello? All right, so if you could look at here, it's a budding cell. This is like something that would stand out for Candida albicans. It would have these budding cells coming out of it. All right, this is as well important. And this one, the other way, the way like you're gonna see it form in chains, like these little coins and chains, it's called the lactophenol cotton blue. Okay, this is the stain that you would use for looking at it microscopically. Okay, now honestly, I've only seen this one being repeated in the past. I've never seen those in the past. The cyani, just to be on the safe side, this is for dermatophytes. This is for Candida mostly, and we use in general lactophenol cotton blue. Okay, but this slide was very much ignored in the past years until last year when they brought a question from this slide. So we have this thing called the germ tube test. Okay. Germ tube test, all right, is basically a rapid screening test for Candida albicans. So you will see these Candida cells and forming germ tubes. If you have a yeast cell with a germ tube, this is positive germ tube test, okay, which indicates that there is Candida albicans. You have to know this. This was asked last year. All right, so you need to know that there's 0.5 milliliter of serum containing 0.5% glucose, all right, incubated at 37 degrees Celsius for 2 to 3 hours. You have to know this. This was repeated last year, okay? Germ tubes are diagnostic for Candida albicans. And this is high yield. You have to know this. Then we move to Aspergillus. Aspergillus species, again, opportunistic species. Usually they do the infection is usually in the lungs, respiratory system. So they usually take samples from the sputum or from like washing the trachea, or even aspirates from patients with pulmonary disease. And this is how it's gonna look, all right, when they grow it on an agar. So this is like at the beginning, and it later on, like after you leave it for like a while, it should look like this, like this whitish out layer and these dark dots right here. This is how it's supposed to look when you leave it for like a good amount of time. Okay, this is something that you want to know. Moving on, we have this thing called mucor, or black or bread mold, as you can see. طيب. This mold is found everywhere. Okay, ماشي, and it causes this thing called mucormycosis. Muco means any mucus, the mucocutaneous layer. So it can affect your eyes, it can affect the lung, it can affect the skin, okay, or even like normal mucous membrane inside the body. It has very rapid growth, okay, and is white to gray and turns to brown when the culture becomes old. So you can see, uh, you can tell like this is a bit old because it turns in just shwy browny, yani. All right, and look, it's hyphae. It's a non-septate hyphae, meaning that they're not branching off each other as like this one. This is a septate hyphae. They're branching off each other. Okay. This is how you want to know. So if you get this one, very I'd say very ugly, blurry, whitish gray, okay, agar, this is mucor for sure. Okay. So we're done with micro. So do you guys have any questions about anything that I've went through? Well, shall we proceed?
Um, I have a question, and obviously it
Sure.
really like depends like it might change like every year, but do you think the OSPE from your batch was mainly like parasites, protozoa, more than bacteria, or did you notice that there are a lot of questions from bacteria, like for the OSPE specifically?
I think it's equal. I think it was equal. But the shock for us last, I mean, 2 years ago was, uh, yani, it was just very like, the way they tested you, okay? They used to like the figures, the choice of figures were amazing actually, and they almost like looked way similar. That's why it was just a bit difficult. But
Okay.
otherwise, it was pure memorizations, honestly, and I think it was equal. I think it was fair. I think our exam was fair. So yeah.
Um,
Um,
yeah, I was
yeah.
actually about to ask you, like, did you guys like saw the same images that they show you in the lab, or like did they give you like brand new, never seen before like pictures?
Uh, Actually, it's half half. Sometimes there will be questions with exactly copy paste pictures from the lab material. Sometimes they're first time figures that we see. And this is
Okay.
why sometimes it's difficult because any it could be like a gram negative, for example, but maybe it's way darker because of the source they got it from. So maybe this will be mistaken. Oh, we're gonna write it's gram positive. This is why it's very important to read the question, very important to know how the disease presents, because sometimes they would write that in the question. Or
Okay.
sometimes they'll be like, what can you see? You know? Yeah, that's
And
it. That's it. Yeah,
yeah,
go ahead.
one last question. Did you guys had like 15 stations as well, or did you have more?
Hmm. I don't remember actually. I think we had maybe 16 somewhere there, or maybe 20, between 15 to 20. I don't really remember, honestly. It was or somewhere between 15 to 20. But sometimes, like some questions can be two questions, like part A, part B. Okay.
Oh,
So yeah, this you have to
thank you
know
so
your figures. يعني حرام تفوتوا الأوس بصراحة POD و 5 credit hour block صح؟ يعني or is it
Six.
six? Six
Six.
credit hours. Yeah, you have to memorize these pictures. يعني حرام إنكم يعني you don't. طيب. All right. So moving on, I'm gonna
Doctor,
start with Yeah, please go ahead.
Sorry, was there any question in your guys' OSPE where they had a picture and they asked a pharma question? Like drugs?
I'm coming to it. I'm not done.
No, because I heard last year they said that they integrated it. So they added pharma and micro together in the labs.
Pharma was asked ever since POD started, I believe. Like, uh, there's one pharma lab, all right. Uh, you know, they get questions from that pharma lab only. And yeah. So, but I'm gonna go over all the concepts right now. I'm not done with the presentation. I'm only done with micro section. All right. So I'm gonna just go through them. Don't worry. طيب, let's do immunology. Okay, immunology يعني صراحة one question would come or two questions, maximum two, I don't think more. Okay, very simple concept that you need to understand antibody antigen complexes. يعني كتير مهم, very important. So simple, antibody binds to an antigen, will give you an antigen antibody complex. Okay, this is this is the fundamentals of whatever I'm going to discuss right now. First thing is the latex agglutination test. So this is usually a test, a very rapid test, by the way. Okay, you would have the antibodies for example, the test itself, and they would put the antigens for maybe your blood sample, all right? And then if they bind, this will form agglutination right here. So you can see this is negative, this is positive. Hello? Usually, this is used in clinics for rapid analysis to identify what blood group you are, okay? Or sometimes there are certain infections, certain antibodies they want to test. So this is effective and very fast, very convenient. All right? Moving on. So we have two things. We have single immunodiffusion and we have double immunodiffusion. So let's start with the single immunodiffusion first. So this is a simple agar gel where the antibody is diffused in the agar. And then there's a well cut where they put the antigens in it. All right. And it's supposed, all right, they're supposed to like precipitate when they, form the antigen antibody complexes. Hello. And, they would form the string-like structure. So you want to know, all right, and this is a fact, and this is very important. The diameter of the ring is directly proportional with the concentration of the antigen. Okay, so the more antigen is there, the larger the ring diameter would be. You know what I mean? Oh, yeah. So diameter of the ring is directly proportional with the concentration of the antigen. This is the takeaway from this slide. And you want to know this is called immunodiffusion. Single immunodiffusion. Okay? These two things. Simple, clear, do not overthink it. خلاص, you see this? This is the only thing you want to know from the slide. Double immunodiffusion, this is where it gets شوي tricky, okay? So it's called, I don't know how they spell this, Auchterloni, whatever. So why is it double? Let me explain to you. So, we have these well cuts of I believe, antibodies, and there are like as well antigens here, like within the agar. What happens if the antigens and the antibodies like meet up, all right, and they form their complexes, it's gonna be what? It's gonna form this precipitate right here. Now, if the precipitate was shaped in this way, okay, this means that okay, this you need the antigens and the antibodies follow a certain pattern of identity. Like this is خلاص, this is known for disease X, okay, or like antigen antibody X. Okay, sometimes there's pattern of partial identity. It looks like this. All right. Sometimes it looks like a cross. The only reason I added this image here is to tell you what normal looks like and what abnormal looks like. So this is normal. Okay, so you just want to know this is a double diffusion. Okay, like it's supposed to form this like arc-like structure here. So when you see this, this is called double immunodiffusion. Hello? This is the only thing you want to know. You don't want to dig into more information. All right. So, but the reason this precipitate formed is the antigen antibody complexes. Okay. So a question would come be like, so name this test. Okay, double immunodiffusion. Why there's a precipitate? Because of the antigen antibody complexes. Hello? Everything's clear? So, ELISA. ELISA, this is very high yield. Okay. Usually they would tell you that, oh, this pregnant woman, she used the pregnancy test and she found out that she's pregnant. What kind of immunological test was that, or like what kind of technology is that? It's ELISA, specifically sandwich ELISA. So let's talk more about indirect ELISA and sandwich ELISA. So indirect ELISA, so you have these antigens right there. Okay, you would add the antibody. Cool. When you add the first antibody and then wash it, all these extra antibodies that were not bind, خلاص, are gone. Only the complexes remain. طيب. Now you want to make them visible. Okay, you're gonna add the secondary antibody. Usually they have these fluorescent abilities, or like, so they would be able to like show colors and stuff. So this is what happens. Now, why do we do this? The reason for indirect ELISA is to measure the antibodies used for these antigens. But when you want to measure the antigen, you use sandwich ELISA, which is exactly this. So you have here the antibodies instead. All right. And then, so let's say, for example, these antibodies are waiting to, for example, catch the, like, you know, the thing that indicates that the female is pregnant. Like, for example, let's say beta HCG from the urine, for example. So these antigens are going to be bound here, right? Then it's going to be washed, and then another detection antibody will appear, which is why you get these two colors. Okay, you get this color first, and then you can get this color again. Like, this is sandwich ELISA. You're measuring the antigen, okay, in sandwich ELISA. In indirect ELISA, you're measuring the antibodies. So if the question tells you, this microbiologist, immunologist, whatever, okay, was trying to measure the antibodies to antigen X, okay, which of the following, which kind of ELISA did he use? Indirect ELISA. You need to know this one. The doctor could literally snip this and put it in your exam. So you need to know this figure, okay? It's very important. So indirect ELISA to measure antibodies. Sandwich ELISA to measure the antigens. Is that clear? Hopefully. And
Yes.
always remember the pregnancy test. Pregnancy test for sandwich ELISA. But T cell activity and number of cells. I've seen only one question, I think, in the entire past, if I'm not mistaken. The question was, how would you assess, all right, the T cell activity and number of cells? The answer was flow cytometry and ELISpot assay, okay, which we're going to talk about. So flow cytometry is very simple. Now, each T cell produce different kinds of cytokines, different kinds of transcriptive factors, right? So these factors and these cytokines need to be mediated to be to be able to be fluorescented by certain antibodies. So this is called, for them to be permeabilized, all right, to allow entry of the fluorescent labeled monoclonal antibody, and this happens right here within the flow cytometry. So the entire idea that you want to know, I don't want to confuse you, that they can tell, okay, the kind of cytokines and the kind of transcription factors released by this T cell. So let's say, T helper, T helper 1 cells, for example, they release cytokine X, transcription factor Y and Z. So when they detect transcription factor Y, Z, and cytokine X, for example, they're going to tell that this is TH1. You get it? This is for flow cytometry. Simple, very clear, very straightforward. ELISpot assay. Usually, what happens is that they would have like a plate coated with antibodies of a certain cytokine or certain transcriptive factor. So, for example, here we have interferon gamma. All right. So this plate is coated with anti-interferon gamma antibodies, and then they're gonna get the cell from the blood. So let's say they got this leukocyte, and they're going to add stimulants to make it to release, um, to release this interferon gamma. So for example, they can add epitopes from certain microbes, okay? طيب. So these antigens that are gonna bind أصلا to the leukocyte are gonna stimulate its production of interferon gamma. Now those cells that are gonna produce interferon gamma are gonna be bound to the interferon gamma antibodies, literally right here. Okay, and then what's gonna happen? We wash it off after like 48 hours. After we wash it off, you're gonna add um like an enzyme labeled secondary antibody, okay, just for labeling purposes. Okay, and then you're gonna incubate it with something to give it like color as well, like alkaline phosphatase conjugated streptozan. You don't need to know this. Okay, and then lastly, you're gonna get these spots. What does this spot mean? This spot indicates the number of interferon gamma producing cells. So each spot is going to tell you that this cell produced interferon gamma, that cell produced interferon gamma, and so on. This indicates the T cell activity as well as number of cells. So ELISpot assay and flow cytometry, both of them will analyze T cell activity and number of cells. Okay, so don't overthink it. Inshallah, it's going to be this simple. This I've explained the procedures just in case. All right. Now I'm done with immunology. We're going to do pharmacology. And pharma is very short, but straight tricky. So I want you to really pay attention to what I'm saying. And if you have questions, please, I'm willing to answer them. Let know it's one question every year, and it's unfair that you solve it wrong. So, sorry. So, are you familiar with these terminologies? Full agonist, partial agonist, antagonists, all those. Do you have any questions in this slide from these different terminologies?
No, it's clear.
Okay. Right. Let's move on. So, this is high yield. So, this is an agonist. Now, we know what's an agonist, like something that binds to a receptor and uh, produces that exact function that needs to be produced. طيب? So, we have two terminologies that I want you to keep in mind. Maximal efficacy, okay? So maximal efficacy is one, and the other one is potency. So when I tell you what happened here, what got affected, potency or efficacy? Can someone like give me a fair guess?
Potency.
Potency. You're correct. Let me tell you why. So first of all, we added a competitive antagonist. Competitive antagonist meaning that they're both competing to the same receptor, right? It's just a matter of who binds first. So the idea here is, how would you know what got really affected? So the efficacy, okay, you check where it lastly ended. So this is the maximal efficacy. As you can see, in both lines, it ended at 100%. So efficacy is undisturbed. But the potency did get disturbed. Why? Because the the half of the maximum efficacy, which is 50%, got changed. So this, this combination is less potent. Okay. Why less potent? So what's the idea behind potency? The idea is this. If the EC50, okay, is low, طيب, this means it's more potent. يعني it can act it can do that like the 50% of the job at less concentration. And that's amazing. Okay. So this is increased potency. So if the curve shifts to the right, potency decreases because the 50% response you need to get are being achieved at higher levels, higher doses. And that's not something that you want, speaking from a pharmacological point of view. You don't want to increase doses, more side effects and that. So the less the dose to get 50% of the response, the more potent is the drug. This is important for you guys to know. Okay, this is a possible question. Okay? So is this clear?
Yes.
Alhamdulillah. Like, let's solve this question together. So let me just change this to a pen. Now I want you guys to find the EC50. So what's the first step I'm gonna do?
I guess look at the point where 50% is achieved.
So first, I'm gonna look at the maximal force. What's the maximal force here? It's gonna be 14. Okay, this is the maximal force. How do I know this? This is where the curve became stationary. Plus, it's not going up, not going down. حلو? Then so it's 14, right? What I'm gonna do is I'm gonna take half of it, and that's gonna be what? 7. I'm gonna look for 7, which is gonna be somewhere here. Okay, sorry for the missing line. So whatever I get on the graph right here, that's gonna be my EC50. You get it? So first step, you want to look for the maximal efficacy, which is 40. You half it, which becomes 7, and then you trace it on the map, and whatever concentration you get here is going to be your EC50. All right? Is that inshallah understandable?
Yeah.
Okay.
Yes.
This came last year. Danny, I think it's going to come again this year, inshallah. It's very easy for you guys to solve, inshallah.
Did they just ask like calculate the EC50?
Yes, they just asked, what is the EC50? All right, like, let's do this one. Okay. So this is they're adding ascending doses of acetylcholine, also a constant dose of X. All right, record was responded. Smooth muscles did increasing dose, whatever. So can you now tell me this figure? So this is the normal one. Let's say this is X, and let's say this became Y. What happened here?
Competitive antagonist was added.
Okay, so when you want to comment on the figure, you want to comment on
The shift in EC50.
shift and potency and efficacy. So
No, only in potency وشوي efficacy.
yeah, but for your sake, to be able to understand this is competitive antagonist, you need to look at the efficacy and potency. Always look at both of them. This can guide you. Okay, so what happens here? Efficacy is unchanged. They're almost the same. Okay, somewhere right there. So efficacy is unchanged. Potency definitely did change because there's a right shift, right? There's a right shift that's happening. So potency decreased definitely. Okay, so the effect of adding X is that it is a competitive antagonist. Okay, is this clear?
Clear.
Selective antagonism. So, do you know what this terminology mean? Or shall I explain it?
Selective, and it's just selective to one thing.
Okay. Yes. So, let's have the example of acetylcholine and histamine. So, acetylcholine targets, the muscarinic receptors. Histamine targets the H1 receptors. Okay. When I add, so here down below right there, I told you that the antagonist for acetylcholine is atropine. Okay. And the antagonist for histamine is mipyramine. So if I add mipyramine all right, and acetylcholine. Will there be any effect? No, because they're working on different receptors. But if I'm adding acetylcholine and atropine, this is selective antagonism, and it's only going to affect acetylcholine function. So let's take a look at this example. So this is histamine. This is acetylcholine. Hello? Is there any change? Is there any change you can witness? Yes. Because see, there's increase in this, there's a smooth muscle contraction happening right here. So there is a change that we can vividly see. There is a change as well here. See, this was working and suddenly not working. And you can see that it only affected acetylcholine and not histamine. So this is clearly selective antagonism. So what do you think that we could have added to cause this manifestation? Atropine or exactly, atropine. Great. Is it reversible?
Yes.
Yes, it is reversible because the second you stop infusing the patient with atropine, خلاص, everything is gonna come back to normal. Okay. Here I conclude my presentation, Yanni. Thank you so much for tagging along for like 2.5 hours. I wish you all the best. If you can scan the QR code and give a feedback, that would be great. Oh, please let me know if you have any questions. Any questions about the exam? I'm more than happy to entertain them.
I do have.
For the last question, sorry, but will they give us the drugs that have selective antagonism, or do we have to
Yes,
memorize them?
No, you have to memorize them.
It's only these four?
There are only two. So acetylcholine, atropine, histamine, mepyramine. Look, if you actually open the pharmacology lab material, these are the only two drugs that they've been repeatedly using, I believe. And even in the past exams, it's always histamine, acetylcholine, histamine, acetylcholine. So it's better to know them because it has been asked before.
Okay, thank you.
Most welcome. Any other questions?
I do have a question.
Yes.
I always confuse Candida and Staph aureus on colonies. So would they bring a picture and say what is this organism? ولا will be there scenario, a full scenario?
I don't think sometimes they'll just tell you what's this organism. هلا the best way if we go back to Staph aureus صح؟ شوفي, we only grow Staph aureus, the only time Staph aureus is going to be goldish is when you put it on a mannitol salt agar or blood agar. So you will definitely see there's slight pinkish around the agar when it's Staph aureus.
Yes, on mannitol
أما
salt is clear, but
on
on blood agar شوية.
on blood agar, if you look مثلا when we grow Candida, we don't grow it on blood agar. We grow it on SDA. صح؟
اي صح.
So
which is much.
so yeah, so ما في blood خلاص you'll be confident انه Candida albicans. This is one thing. Tiny thing, Candida looks like pearly. Look, تحسيها شوي كذا زي pearl. تمام؟ So this is one of the things that might help you to differentiate. It's a bit pearly in structure. Okay. Oh, there's no blood. This is Candida albicans.
Yeah, when you said there's no blood, it صراحة makes so much sense. Thank you so much.
والpearl كمان شوفي شكلها زي الpearls. You know the pearls?
Yes, زي لولو
Yeah.
Thank you so much. Appreciate it.
You're most welcome, of course. Any other questions? So I think it's a no. Let me check the chat. All right. Thank you so much, everyone. I truly appreciate it. And inshallah, good luck. If you have any questions, these are my contacts at the very end. Oh my God. Sorry. Just a minute. I don't know. Can you see it? My contacts were right there. So yeah. Thank you so much. And I hope you have an amazing exam. Just make sure that you read the question very clearly. And inshallah, all the best. Thank you
يعطيك
so much,
العافية
everyone.
إسماعيل.
Allah يعافيك. Thank you so much for attending.
Welcome, Mullo.
Yeah. Goodbye.